right. Okay. I'm going to share. Wait, Sebastian, quick question. When I share, can you see like the people on the side, or is that just my screen?
Yeah, I can.
Okay. Okay. So we're going to talk about DNA replication. It's a pretty short review because we just did Monday and today's lectures, but hopefully it'll clear up some things if you guys have any questions.
Yeah.
So nucleotide structure. If you guys have taken 151 in the past, I'm sure you've gone over it, but we're just going to go over it a little bit more. All nucleotides consist of a phosphate group, sugar, and a nitrogenous base. That's what makes it a nucleotide. If you see a nucleoside, that's something very different. Don't get them confused. Pointer. There it is. So here you can see this is your phosphate group. Usually on the 5' end, you have this OH group here on the 2. This is RNA, by the way, because RNA has this OH on the 2, whereas your DNA does not have an OH on the 2' carbon. And your nitrogenous base is either going to be your adenine, guanosine, thymine, or cytosine, or uracil in cases of RNA. This is the structure you're going to see for all nucleotides for both RNA and DNA. You have your adenine, your guanosine are purines, and thymine and cytosine are pyrimidines. One way that helped me remember this, it was like a mnemonic from Bio285, but it was angels and gods are purines. So that's kind of what helped me remember that, A and G, pure. Also, I don't know if Laura's gonna really need you to know this, but purines are 2 carbon rings and pyrimidines are only 1 carbon ring. I don't think that's critical to what you need to know, but it's just something to add. Does anyone have any questions on nucleotide structure? No? Okay, cool. Sebastian, you want to do this one or you want me to keep going?
Yeah. Yeah, so like Rachel said, I really like that angels and gods things. That's really cool. Okay, so your adenine binds with the thymine. You guys probably knew that
sorry,
already.
I'm so sorry.
Yeah,
there's a typo on the slide. I'll fix it. Purines are not adenine and thymine. It's adenine and guanosine.
Oh, okay.
Yeah, that's a typo. I'll fix that.
Yeah, you're fine. Thank you. Oh yeah, I was looking at it now and I was like, God does not start with a T.
No, these are your purines, A and G, thymine and cytosine are your pyrimidines.
That's funny. Okay, well, besides like the purines and the pyrimidines, the takeaway from adenine and thymine is that they form 2 hydrogen bonds, while the guanosine and the cytosine form 3 hydrogen bonds. You can see that illustrated on the left-hand side there. A big thing that Laura really accentuated, and I think it was the first or the second lecture, was that they always, or that like DNA forms in a bidirectional manner, which like if you look at the strand on the left, a good way that she described it was the the sugar base has that oxygen that points up, and then on the other strands, the strand on the right, the oxygen on that sugar points down. So that's a good way to remember it. So we've definitely talked about a bunch of different bonds, like ionic bonds and peptide bonds, whatever. There's one more bond that you probably should be familiar with. It's called the phosphodiester bond. And that's just pretty much, I don't know if you can see my cursor, but that phosphate group is pretty much bonding with the hydroxide group that Rachel talked about on the previous slide on the 3' carbon. So the 5' phosphate, again, binds with the 3' hydroxide, and that's what forms the phosphodiester bonds. So any questions about that so far? Anything you want to add, Rachel?
No, that's pretty much it. Then we just had a quick question. Which bond do you think would be harder to break, A and T or G and C?
Yeah, so does anybody have an idea? You could drop it in the chat if you don't want to like turn on your mic or something.
Totally fine.
Hydrogen bonds. So normally if you're going to have any breakage between nucleotides, they're going to want to do it between.
Jessica, you cut out.
Oh, dang it.
No, that's okay.
Just G and C would be harder to break.
Yeah, you're right. Because it has, they have 3 hydrogen bonds where A and T have 2.
Yeah.
I'm also quickly just going to edit this while
Yeah,
we're
no problem.
here because I don't want people to
Yeah, that's fair.
get.
Oh yeah, that came out in the slides that I just sent you, Jessica. So just remember to like cross that part out with the
Oops.
pyridines and pyrimidines. Anyway, yeah, so you guys have probably heard of like the Tata box before. That's where, correct me if I'm wrong, Rachel, that's I think where the polymerase like binds, or that's a bonding site for transcription factors, was it? Or the
Yeah.
polymerase? Something like that. Yeah. So the reason it's binding to the Tata box, which is just a sequence of adenine and the thymines, is because that bond is much easier to break. It's easier to rest on than that triple bonded guanosine and cytosine. So that's like why the Tata section, or like that sequence of AT bonds, is a popular bonding site for polymerases or transcription factors.
I also just want to point out here, you can see that the purines, adenine and guanosine are double carbon rings. So you have 2 carbon rings, and your pyrimidines, thymine and cytosine are 1 singular carbon ring. It's the same with uracil, because uracil is a pyrimidine subsin for thymine and RNA, and that would just be a 1 carbon ring. And the main takeaway really from this slide is just remember that you have that 3' hydroxyl group that makes a phosphodiester bond with the 5' phosphate group. That's pretty important. And in DNA nucleotides, you do not have a hydroxyl group on this 2', it's just carbon. Whereas if we go back to this slide, you see on a RNA nucleotide, you do have this hydroxyl group, but for DNA, you do not. That's pretty important.
For dideoxy, we haven't talked about dideoxyribonucleic acid, but dideoxy just means that it's missing both the 2' and the 3'. That's pretty much like at the end, like at the very, very end of the DNA, when it doesn't want to bind to any more nucleotides. That's where you'll find like dideoxy, and it's just pretty much instead of those 2 hydroxide groups, or that 1 hydroxide group on the 3', you'll see a hydrogen there also. So di meaning 2, dideoxy. So the 2' and the 3' are missing those hydroxide groups. It's a little like not too relevant for the purpose of this class, but just in case it comes up later.
Just those 3' and 2' hydroxyl groups are pretty important for the structure. So speaking of the structure, here we have another diagram. Here you can kind of see the phosphate group, like the molecular structure or the molecular formula of it connecting to that 3' hydroxyl group. But so important things to know about DNA is DNA is polar and negatively charged. So DNA polar, it's not symmetrical, it's different. The 5' end, so up here of DNA, has a free 5' carbon attached to the phosphate group. It's not attached to another nucleotide. So see how it ends here with this phosphate group. And the 3' end of DNA has a free 3' carbon attached to the OH group. So you have your free 5' phosphate and carbon at the 5' end, and then at the 3' end, you're going to have a free hydroxyl group. Another thing to know about DNA is it's double-stranded, right-handed turning. As you guys have probably already have known from your many biology classes before, A always pairs with T, and A pairs with U in RNA, and G always pairs with C. So DNA is coiled, and that's stabilized by van der Waals interactions between, I don't know, is it the nucleotides van der Waals are between? Because it's does like the spiral, and it's very tightly coiled before replication, which we're going to talk about a little bit in a minute. But when it's very tightly coiled, I think the van der Waals interactions are up and down, but I'm not entirely sure. I think they are. But yeah, so this is basically the structure. As you can see, the second, it's anti-parallel. So this 3' matches with the 5', and it's basically, it looks upside down in this diagram, but that's how it orients in order to make that double strand. And then once again, you can see the 2 rings for the purines and the 1 rings for the pyrimidines. Any questions so far? I know you guys have probably talked a lot about this in your previous bio classes, but it's kind of just a review. But yeah, please stop us if you have any questions.
Yeah. Okay, so this image always used to like really mess me up when I first saw this, but pretty much what happens is let me just read the slide real quick. Occurs in two different directions. Okay, yeah, so just to briefly summarize this slide, there's an origin of replication, which you could see by that dashed line. So if you could picture like DNA bound together, like if it looks like a little railroad, right? And there's no replication fork yet, just whatever. At some point at the origin of replication, your helicases are going to bind there. And when they bind there, they're going to go in opposite directions. So that's why at the top of the screen where Rachel's moving like the cursor right now, that's why you have like a leading strand there. But then right below, you have a leading strand that goes in the opposite direction. So don't worry about like everything before that yet. Just know that when the DNA is just bound together, it's not like ready for replication, just kind of looks like that railroad thing. And then when it is time for it to replicate at the origin of replication, those, I believe it's like the MCM to those helicases, they bind there and then they start going in opposite directions. So then the question is, how does it, like you can probably like clearly understand how it's capturing everything or how it's replicating everything in the 5 to 3 prime, but then what happens everything before that? And the answer to that is Okazaki fragments, which we'll talk about in a second. But again, like just visually, this slide is always like a little confusing. At least it was pretty confusing for me the first time I saw it. So do you have any questions about like really what you're looking at, or why does it look the way it does?
Just to be clear, like when the MCM complex binds at that origin of replication, yeah, like right where Rachel's circling, are they going to the directions to start their leading strands, meaning that one's going to be at the top going down,
Like
and
this
then
way?
another one's, yeah, and then the
And
other
then
one's going to be the other one for the leading strand? Is that where they'll be located?
Yeah.
Exactly.
Okay. Okay. And then so then if they're located there, I'm assuming like, do they ever move, or do they stay right there at the origin of replication? And if so, like how do they how are they incorporated with lagging strand? I know we haven't gotten there yet. I'm just curious, like based on like the diagram, how that operates.
Yeah, they move. Helicases or the MCM complex have an ATPase activity, which we'll get to we have like a whole protein chart with all the proteins that happen at the origin of replication. But yeah, we will talk about that. But yeah, the helicase moves along with ATPase activity, which is the MCM complex. I'm not sure how the sliding clamp moves because that's what holds the DNA polymerase in place, but the DNA polymerase itself, when it's replicating here on the leading strands, has its own activity that allows it to move.
Yeah. DNA polymerase, but the helicases though, I don't think do they.
The helicase yes, they do.
They'll move with them.
Yeah. So the helicase
Yeah,
moves,
that makes sense because they okay.
right. And we'll
Yeah.
talk about it, but it's basically just ATPase activity. It hydrolyzes ATP and that's what allows it to move along. But the summary slide at the
And
end.
the lagging.
Yeah,
Yes.
for both.
The
for both.
lagging strand.
For so the lagging strand is the Okazaki fragments. It does the same thing. Sebastian, correct me if I'm wrong also, but I'm pretty sure that the helicase moves with that ATPase activity on the lagging strands also. It just has to pick it up because, well, you have to remember that the helicase is just unwinding your DNA.
Right. So then is there 4 helicases then?
No, so the helicases bind at the origin of replication, and each helicase goes in the opposite direction, and that's what's causing like that fork. But as soon as the helicase has unbound that region and it's like loose, I guess, that gives it an opportunity for the DNA polymerase to bind there where the helicase used to be. So the polymerase is pretty much chasing the helicase.
Yeah.
And that's the helicases are what, there's only 2 helicases. There's one like on that top strand and there's one on the bottom strand. And each of those, it's what's making the fork. It's what's like expanding the fork pretty much.
Okay.
But the DNA polymerase is trailing like very close behind it, just leading along.
So would we assume, and like maybe this is wrong, because I know the helicase is splitting this 2 strands. So does that mean that it's large enough that like it's going through both strands? So where that on either end, like where like you start to see the railroad track, Is that where helicases would be? And it's starting to go and it's causing those bubbles. And then right behind it is the following DNA polymerase. Is that more what it would be like?
Yeah, don't think so. Yeah, so don't think of the helicase as
On
in
like a single strand.
yeah, so don't think of the helicase as in like the leading and lagging strand. It's just opening this bubble to make it available for that DNA polymerase. So you have one helicase moving this way, which is why the DNA polymerase is allowed to come in, and then you'd have one here moving this way, which allows it, which is why it's bidirectional. So it's not in terms of the leading and lagging strand, because the helicase doesn't have anything to do with replicating the actual DNA, which is what these strands are doing. It's just opening this double-stranded DNA to make it single-stranded. Does that make sense? Okay. Yeah. And another important thing about this slide is just that we'll get to it also, but DNA polymerase synthesizes DNA only in the 5' to 3' direction, which is why we get these lagging strands. But we have a whole slide on that, so we'll get to it. But so you just did the last one, right, Sebastian? Last slide.
Yeah.
Yeah. Okay. So the addition of nucleotides, nucleotide synthesis always goes, like I just said, from the 5' to 3' direction. The 3' hydroxyl group always gets the next nucleotide. So down here you have this end of the strand, this 3' end. This is where this new nucleotide will come in and bind with its phosphate group will bind to the hydroxyl group of the 3' end. So that's where that 5' phosphate makes that phosphodiester bond here. So the reason this is important is because DNA polymerase needs an existing template before adding nucleotides, which is what those RNA primers are for. So I just lost my train of thought, sorry. So yeah, so the RNA, so it's called primase, but it puts down those RNA primers when we get replication, and that DNA polymerase needs that RNA primer in order to start adding nucleotides to the existing template. So the reason that we get a lagging strand is because polymerases move in opposite directions. So if we're trying to get all of it replicated, how does that happen? We get that lagging strand because we have to go that 3', 5' direction. And if you look here on this slide, we start here. So we have a 3'. So we're going this way, but we can't synthesize 3'. So I'm always so bad at explaining this. So okay. You have your 3' and your 5'. Here it's easy because you can just add them on 5' to 3'. But here it's not so easy because you look at it here, you're having your 3'. This is where your 3' end is, and that's why this says 5', and then you need to go to 3'. Sebastian, if you want to help at all. I always get so bad at explaining this.
No, you're fine. Yeah, so like Rachel said, at the origin, like, okay, the helicases are splitting, whatever, but for the DNA polymerase, the leading strand, obviously it just needs, the primase needs to put down like a little template. That template is super important because otherwise the DNA polymerase can't catalyze that reaction. It needs like a template to write on, right? It's like trying to write on a paper, but you don't have paper. So that's pretty tough. So the DNA on the polymerase on the leading strand is just going to do its thing because it's going in the direction that allows the phosphate to attack or it's going in the direction that allows the hydroxyl group to accept the phosphate group. If you try going in the opposite way, that's why that reaction won't catalyze. The hydroxide needs to accept the phosphate group. And if you go in the opposite way, it would be like the phosphate group trying to accept the hydroxide, like that bonding site just won't work. So what happens is that you have, again, those primases that are putting down templates before that. So again, we know that the DNA polymerase is from right there where Rachel's cursor is. We know that it's going to be traveling to the right, but the primases, after it's done making like that primase for the leading strand, it's going to step back a little bit. It's going to make its own template. Laura said it was like 100 base pairs long or something like that. I don't know, that's probably not super important, but whatever. It's going to put down like another template. And once that template is made, it's going to jump back like another, I don't know how many base pairs, like 1,000. I don't know how many actually, but it's going to jump back. and it's going to put another primer, it's going to jump back again, and that cycle just keeps going for the rest of the DNA. Once all of those primers are there, those Okazaki fragments are there, then a different DNA polymerase, so not the one making the leading strand, the DNA polymerase on the leading strand, that's doing its own thing. As far as I know, there's only like one doing the leading strand. That dude is just going to keep doing his thing. But you do need more. You could probably imagine that you're going to need like a lot more DNA polymerases for. It's not that there's one polymerase for each Okazaki fragment, but you're definitely going to need more, because if you have a bunch of Okazaki fragments, to only have one polymerase responsible for polymerizing each Okazaki fragment, I'm sure you can imagine how that could take like a really long time. So there's a lot more polymerases responsible for generating or polymerating that strand from the Okazaki fragments. Does that make sense?
Yeah, I had to think about it for a second, but if you start here, the way that it works for that, why that 5' to 3' doesn't work is because you can't go this, you can't go, so see where my cursor is, you can't go to the left. You have to keep going this right way. My thoughts were all jumbled. But yeah, so that's basically how it works. I think eukaryotes have 3 DNA polymerases, might be more, but I know there's definitely 3. But we'll get into that, I think, a little bit. So a quick question that we have is, the DNA polymerase moves along the template strand in the blank direction, and the daughter strand is formed in the blank direction.
Yeah.
So
what do you guys think? Again, you could drop it in the chat, or you could say it out loud. It might take, I understand that this one might take like a little bit to think about, so take your time. So many papers today.
what do you guys think?
If you guys would rather like we explain it, that's
Yeah, we can
totally
just go
fine.
through it.
Yeah.
Keep thinking 5', 3' for both, but like I just have a hunch that that's not right. Like there's some I remember learning at one point there's something that's a 3' to 5'. I'm not sure if this is it, but so maybe explaining that might be helpful.
Yeah, I think you are thinking of the right thing. So the DNA polymerase, so careful with the language here. The DNA polymerase, like the protein itself, is traveling along in the 3' to 5' direction on the template strand. So it's moving in the 3' to 5' direction. However, it's adding the nucleotides, it's adding the newly synthesized strand in the 5' to 3' direction. So although the polymerase is adding nucleotides in the 5' to 3' direction, it's physically traveling along the template strand in the 3' to 5' direction. So the answer here would be D. The polymerase moves along the template in the 3' to 5' direction. However, it's forming the new strand in the 5' to 3'. That's the important part.
Yeah, the strand always forms the 5' to 3'.
Right. Does that make sense?
Cool. So we kind of talked about this already, but here's some more lagging strand.
Is this my slide?
Yeah. No, sorry, you're good.
Yeah, no, you're fine. Yeah, so okay, so we talked about how the polymerase polymerizes the leading strand. So we'll talk a little bit about how the lagging strand works. So again, primers are still required. Again, that's like trying to write on a paper where you don't have a paper. So that's pretty difficult. Primers are always required when you're making some kind of strand polymerization. Those fragments that you could see like in green, well, okay, like right there at the line where it says origin, the 5' to 3' direction, that like one green primer before it goes into the main leading strand, that's just like the regular primer. But the primers before that one, those are called Okazaki fragments because of that power couple that like discovered these. So again, we already talked about it a little bit. The primases are going to jump back a little bit. They're going to make a little template. They're going to jump back again. I don't know how many nucleotides, like 1,000, I want to say, I don't really know, but they're going to jump back more. They're going to make another another primer, jump back, another primer. And then those other DNA polymerases are going to come in and they're going to, from that primer the primase put down, they're again going to start generating nucleotides or generating the newly synthesized strand, also in the 5' to 3' direction. Again, it has to be taken into part. It has to be in increments like that because of everything we just talked about. But an important protein to keep in mind is there's this protein called the ligase, which the ligase is pretty much just responsible for creating that phosphodiester bond. So if you look really carefully, I know this is just like a cartoon, so it's like a little harder to visualize, but like right there where Rachel's cursor is, you have one nucleotide right before, so there's actually like a space. There's a space before that last nucleotide that the primer made and the next Okazaki fragment. That the polymerase does not catalyze that phosphodiester bond. So there's just like an empty space sitting there. And obviously the cell wouldn't want that, DNA wouldn't want that. It wants everything to be cohesively bonded. So there's a protein called ligase that pretty much catalyzes that bond to fuse together the newly synthesized daughter strand from the Okazaki fragment onto the Okazaki fragment that it's right next to or that it's approaching. So in other words, the ligase just pretty much fuses all of the Okazaki fragments and the newly synthesized sister strands that it's attached to or that's polymerized out of it. It just fuses all of those together by catalyzing a phosphodiester bond.
It's the glue. The
Yeah.
ligase is the glue. Do you guys have any questions on that? If here you can kind of see the origin, you have this going in this direction and going in this direction, which is why the DNA polymerase can't go to the right here where my cursor is. It has to keep going left, but it can't because it has to go in that 5' to 3'. Well, it moves in the 3' to 5' direction, but it has to add in the 5' to 3' direction, which it wouldn't be able to do if it went right here. But here it can because you're on the other strand and it can't go left. So that's the whole basis of it. So this is our summary slide, basically, of all the proteins that you need to know at the replication fork. First, we have the helicase, which we talked a little bit about. It unwinds. So dsDNA just means double-stranded DNA, and ssDNA is single-stranded DNA. So dsDNA, it unwinds it, and which requires the breaking of hydrogen bonds. Because if you remember from our structure slides, you have those hydrogen bonds between each base pair. And so the way it moves along is an ATPase activity. It uses the hydrolysis of ATP to move along each strand. In E. coli, it's called the DNA B, which I don't think you really need to know. She's going to ask you that. She'll add that it's the helicase. And in eukaryotes, it's the MCM complex, which we kind of talked about in the previous slides. Then you have your DNA polymerase. The technical name is genomic DNA polymerase. In E. coli, it's DNA polymerase 3, and prokaryotes, it's DNA polymerase epsilon and delta. These are just kind of, they all do the same thing. So all of these all do this function. They synthesize the new strand of DNA in the 5' to 3' direction. Eukaryotes have several different DNA polymerases. I don't think you need to know any ones other than genomic DNA polymerase because that's the important one for the course of this class. But something you should know is that DNA polymerase 3 and all of its sister polymerases have a very accurate proofreading ability. So if they catch that that they're putting in the wrong base pair, so say it put like an A with a C, it's able to go back and fix that before it moves on, which is important for, as we talked about, the cell cycle. You don't want mutations in your DNA that could lead to cancer or disease cells. But when you do get those, so sometimes it's not 100%, so sometimes it will miss a few base pairs if it makes a mistake, which is why we have those checkpoints in the cell cycle to go back and double check. So the sliding clamp, she talked a little bit about it. It basically just holds the DNA polymerase in place in the DNA so it doesn't fall off. And the sliding clamp loader is exactly what it sounds like. It puts the sliding clamp on the DNA polymerase. It is an ATPase protein, which is just probably important to know. That's how it does its job, is through its ATPase activity. And then primase, we also talked a little bit about. It adds the RNA primers, which DNA polymerase can't add nucleotides without this primer. And then the single-stranded DNA binding protein is very important because after helicase unwinds your double-stranded DNA, single-stranded DNA is your body or your cells like to attack it because you're not supposed to have it. So they sense it as foreign. So by binding to the single-stranded DNA, it protects it from degradation because it's a very unstable molecule. And both strands of the single-stranded DNA are going to want to reconnect with each other, which if we're trying to replicate, we can't have that. We need that bubble. We need them apart if we're trying to replicate. So the single-stranded DNA binding protein not only protects them from degradation, but also keeps them from connecting to each other. And that's it. That's all we have. It's basically everything she covered today. If you guys have any questions, you can obviously let us know.
I have a question. Did you have to know the DNAB for the MCAT?
Yes and no. Like, let me show, since you guys are here, you can see my MCAT notes on this topic.
About MCAT, and it was like Sebastian was reading my mind because
oh, don't worry. This is my entire MCAT folder. You guys can see this, right?
Are you studying for it soon, Jessica?
Studying for it. So I graduate this spring, and I'm gonna be studying for it over the summer. And I've had like a few meetings with Kathy, because she's my pre-med advisor.
Kathy, I love Kathy.
Yeah, great. It's just like, it's funny because like every time I ask her and it's like getting closer and closer, she's like, you don't need to worry about it yet. You don't need to worry about it yet. And I'm like, but I need to know.
Yeah.
I have no idea what to do for my study plan. I was considering doing like a prep course. I
That's
know
what
they're
I
like
did.
not expensive. So you did the prep course? See,
Yeah.
She told me like not to do it and invest my money. And like, I know it's super expensive, but like I've been setting aside a lot of money for it just in case I decided to. What did you use? Did you do like
I used the Princeton review. It was pretty expensive, but they gave me all the books. They had like online lectures that they had live lectures, and then they had pre-recording lectures. So I kind of watched both. And then they had, so if you, right here, they had these like FSQ drills, because that's what all the multiple, basically like made after all the multiple choice questions that you could see on the MCAT. So like if I go to genetics, like these were just, so they gave you in-depth explanations for every answer. So this was me going through each drill and writing down the ones that either I just wanted to go over or that I got wrong. And I wrote down all their explanations because they like do pretty in-depth explanations. And I put like the topic here. You can see like mitosis, meiosis, and like certain things that they highlight, and then they kind of like take all the data from the things you got wrong, and they tell you which subjects you need more work on and which ones you're pretty good at. Another thing about it is their practice exams were harder than the actual exam, and they do that on purpose so that when you take the actual exam, it seems easier, which it did for me with some subjects. So like I did on all my practice tests, my chem and physics score was the lowest, but on the actual exam, it was my highest score. But then for biology, my exam's biology section was ridiculously hard, and it ended up being my lowest score when biology used to be my highest score. But I really liked how they did that, like how they made their exams and their practice questions a lot harder, because it made the actual exam easier.
Practice questions, because I know like the advisors, like they just emphasize like practice more so than the content review. So I'm assuming like Princeton Review does a good job of like making sure, like is it very content heavy, or would you say that they go over content based on practice questions?
So if I show you here, so my MCAT folder, all of this, so this was all content review. Biochem is one of the shorter sections, but like my biology, like all of these folders, like this was all content review that I did through the class and through my own reading of the book, because they would assign like for homework, like do this. Like this was during the class, what I learned. And then I made like tables, because for me, visualizing with tables just works easier. But it was a lot. The good thing though is with the books, so it's a lot of content review. For me, biology was easy, but like for chem and physics, like I hate physics, and like you can see I literally don't have any notes because I did it mostly on like a notebook because it's mostly math. And like orgo was not fun either. But it was just like, for me, the biology was the most because that's the most content heavy, and it kind of was nice to just skim the book a little bit. And then the class also highlights like the big things you should know. So like the reproductive system isn't really on the MCAT. Like there may be 1 or 2 questions, but like in your overall score, like you're gonna need to focus more on molecular biology, microbiology. They love the nervous system and the heart. Like those are their big ones. So like it kind of helped me realize what I needed to spend time on and what I didn't need to spend time on. Another thing is they in their books, they have these practice passages. I don't know why I just put that here. So I only took notes on one of them, but for like fetal development, it's based on, so like the MCAT's set up, I'm sure as you have known, it's multiple choice questions and then it's practice passages with multiple choice questions. For bio and chem, a lot of it is mostly like experiments, but so like this one was basically the topic that the passage was on, and then the questions that they asked about it. And each passage usually only 4 to 6 questions anyways, but their passages, like they had really good explanations for each question, I thought. Another thing though is if you don't want to take the class, so like I took the class and it was like 3 months, and it really forced me to do like 40 hours a week of studying. Like that's why I have all of these notes. But if you don't want to spend that much money, UWorld, here I'll pull it up actually.
UWorld is like a great option, and then
It's
the
great, yeah. So
AAMC like books to do practice problems in, I know
Yeah.
are really good.
Right.
Like I was considering doing it. Like I know that Princeton Review has this deal where it's like if you don't get like a 510, depending on which one you choose, but like if you don't get like a 510 or 515 plus, you get your money
Yeah.
back or something.
Yeah.
Is it like, did you feel like, do you need to take the test again, or did you feel that the class was helpful enough that you were happy with your score?
So I am happy with my score. I don't have to retake the MCAT, thank God. But I used both UWorld and the course. So UWorld's like $200 for like, I think it's 3 months or
I
something.
plan on doing both. I just didn't know if like the class was like a decent
So
investment.
the class really helped me prepare for psych and soc, like that section, and more bio and biochem for me, and orgo actually. But for physics and chem, which is their own section, I don't know if it was either it's the same thing problem I had with professors here, whereas like they know so much about it, they don't know how to explain it in simpler terms. But I felt like for me, the chem and physics portion of the course was a little too fast, and we spent a lot of time on like really complicated subjects that I felt like I needed to know like the basis first. So that's what I used UWorld for, because it kind of just generated like constant questions. So I was just like drilling questions, like drilling physics for like 2 hours, and then I do chem for 2 hours, because it would just constantly give you any types of questions you would see on the exam. Whereas like the course was more like, today we're going to talk about, the Bohr model, and it was like 3 hours of that, so it kind of felt, for those courses, I felt like it was a waste of time. But for psych, soc, biochem, and orgo, it was very, very helpful, I thought, at least.
Wasn't the Bohr model like proven to be incorrect?
Yes.
Like years later. Okay.
Yeah, even useful. The only thing that I remember from like Gen Chem that was actually like considered like useful was like the Lewis dot structure thing.
Yeah.
Because we had to use it for like, I can't even remember what. All I know is like, yeah, it was important. That's what my memory is. It's like, thank you, college. I've learned so much, so I'm gonna have to like reteach my entire like.
No, exactly. And that's basically what I did with Gen Chem. I had to relearn. I was like, I don't remember anything about any. Luckily, I had just taken biochem the semester before, so it was kind of fresh in my mind. But like the Gen Chem, I was like,
That's why honestly, like I'm so thankful for the LRC and working as an SI for Gen Chem. I worked
Yeah.
in Gen Chem 112 for 3 semesters.
Oh,
So I have a pretty good,
3.
yeah, 3 semesters straight. And it was like, I personally, like, I love chemistry. That's like my thing. But if I could recommend anything, I'd recommend TAing a lot, especially for a course like this, where it's very like molecular bio heavy. If you can, definitely try to apply to the LRC. And this goes out to like people watching the recording as well. If any of you are pre-med or interested in studying, I definitely recommend try applying to the LRC, because you do get to retake these courses. Like I'm forcing myself to SI for Orgo 2 next semester, even though I really do not want to, because making those worksheets, it's
Oh God.
the bane of my existence sometimes. But you know, I don't remember anything from Orgo 2, so I have to force myself to SI for Orgo 2. The Gen Chems, I feel like I'll be okay, because I did that for a few semesters. I'm TAing now. I TA'd last semester for genetics. I'm doing this, whatever class we're in now, obviously.
Genetics.
I'm gonna force myself to TA for something else, you know, bio-related. So if I could recommend anything to anyone with the MCAT in their horizon, just definitely seek out TA LRC opportunities.
Yeah.
Obviously, it's good for your resume, but it's also like good for you as well, just so
Yeah.
you could recap on all those things.
Best things. Like it's funny because I actually like emailed Laura about like TA, like she's full next semester. So I was like, dang, you know, it would have been like great because I love this course and like I love what you guys do. It's like so helpful. Like I literally live and thrive off TA review sessions just because like it's such a great.
Yeah, honestly, it's
I love
forced me to
teaching.
like relearn a lot of things, like DNA replication, for example, or
What a cell is. Just kidding.
yeah, like the
But
cell
like
cycle.
honestly, you forget and you're like, oh wait, this is new. And then like you ask like all these probably the same questions you asked like semesters ago. You know, like I probably learned replication and like, oh, what is like a eukaryotic cell versus like prokaryotic cell? And yet every time when I relearn it in a class, I'm always like, oh, like I remember this now. So I kind of like the repetition because like you guys say, like it just kind of like stays in your brain.
Also, learning it in a format where you need to teach it to someone helps you understand it better than if you're just trying to memorize it yourself. If you're like, oh, I have to teach someone this, it definitely helps.
Right. Learning how to like teach something is so different from like you learning it yourself. You know, that used to happen in my sessions all the time. Like I would understand it fine, but when it's time for me to like communicate it, I'd be like, I don't really know how to like explain this. So yeah, practicing like, and obviously like learning it, but like how Rachel said, practicing how to explain it to someone else definitely helps out so much.
Why I was considering doing the class because I'm a discussion-based person. Like I work really well when I am able to go through things with people. Like that's one of the main reasons why I love going to TA sessions or help sessions and stuff. And like I'll be like, hey, let's just go over problems. Like it may not be like it's not that I don't know how to do it myself. It's just like being able to talk things out.
Yeah, I definitely agree. But if you guys
Like,
are all,
does it offer that kind of thing for you? Did the class at all?
yeah, I think
Yeah,
it definitely does.
like office hours and stuff.
Yeah. Because you never know what like someone's going to come in and ask, and they may ask you a question. You got to think about it like, oh, like, let me look at my notes and try to figure it out.
People get really creative with their questions too. So they're really going to force you to think about like
I
things.
know.
There's a lot of
sometimes
questions that people ask Laura, you know, I'm like, I honestly just think I'm like, how do you know this? Like, I don't know this. I have no time to know that at all anyway.
Yeah.
So kudos to them. But yeah, taking the MCAT yourself or are you?
No, I'm waiting for like you, I'm waiting for after graduation. Like props to you, Rachel, for balancing schoolwork and job while also studying and like killing that
In
MCAT.
hell.
I dedicated my entire summer to the MCAT. I mean, honestly, COVID kind of helped me out in the sense that I wasn't like distracted by my friends wanting to go out or my boyfriend being like, let's go do this, and my family being like, let's go on vacation. So I literally had nothing else to do other than study. So it kind of worked out. And I also was abroad last semester. So when I was planning on coming back in May and starting my studying like as soon as I got back. But I came back in March. So I just started reading the biology textbook because I was like interested in it. And then like by the time May came when I was like originally supposed to start studying, I'd already finished 3 of the textbooks. And I was like, oh, I'm like ahead of my schedule.
Awesome. Yeah.
So kind of worked out in that sense. But
You and in coupling with like the U world and everything.
Yeah.
it was really good.
I also definitely
Okay.
recommend looking at the MCAT. It sounds kind of funny, but the Reddit MCAT thread, like people post their flashcards. They'll be like, oh, I got like a 520 and like these are my flashcards that I used. Or like people post like study guides that they've made, and like it was like very helpful. It's kind of overwhelming because a lot of people post their scores when they get them, and it's like, oh, like you got a perfect score. That's crazy. But other than that, like it's very like they post a lot of good resources, and like I had like never had a Reddit before that, but like one of my friends was like get it and like look at the MCAT subreddit, because it like they like literally post like so many resources that I would never have known about if I hadn't had it.
I'm sure there's a lot of cool acronyms on that Reddit
Yes.
too.
Yeah, there are. And it's kind of comic relief.
Yeah.
You'd be like, oh, I spent 3 months studying and I got a 490, and it's like, oh no, yeah, I know. This one guy posted it and he was like, oh no. Oh, I'm so sorry. Another big tip, and this is for anyone watching. This is like for anybody who's pre-med watching. Like when you take your first practice MCAT, like don't go in thinking you're gonna get a really good practice score, even if you're like, oh, I took all these classes, because you probably won't. Just for reference, I got a 499 on my first practice test, which like I was disappointed about because I wanted above 500, but then I realized like most people don't get 520s and above. Like it's really only like the top of the top. So it's like, don't be discouraged if your first MCAT is or your first practice test is a 400 or lower or whatever, because mine was 499 and it took weeks to get it above 500. Like it took a while, and then I finally did. But it's not fun. It's not how much you know, it's how well you know how to take the MCAT, which is why it's practice, 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 because it's like, oh, that's a key word that you might have missed. So it's good, but
Yeah.
Future doctors, woo.
I feel like we spent just as much time talking about the MCAT and pre-med stuff as we did like during
I know.
Questions though, like it's so useful because like honestly, like that's what like Kathy was telling me. She's like, oh yeah, we're gonna have an MCAT panel soon, just like go there, you know, maybe they can help inspire you to figure out what you want to do. And I'm like, that's great, but like I want to know now. Like,
Yeah, I know.
I want to figure out what I should do and what's a good resource, because she highly emphasized not doing the class.
Yeah.
You know?
If honestly, I feel like if you have the discipline and you get UWorld and use other resources, you get the AMC books, you don't need the course really. I did it because I knew I would not force myself to get up at 8 a.m. every day and log on to a class. So it gave me that structure that originally helped me. But once I like got into my groove, like I didn't really need the class either when I got to it, like later in my studying. It kind of forced me like at the beginning of my studying to like get up early in the morning, start studying, do like practice questions for like 2 hours. Like that's why I did it, but it is a lot of money, and if you really don't want to spend that money, you don't need to. Like plenty of people score good scores without it, you know.
Like they do content review, because it's like I have no idea how I'd structure content review just based on problems. Like I don't know where to start.
Yeah, it's hard. It's like there's so much, and it's very overwhelming because you're like, oh my god, like they could ask me anything. But like when you go through it, it's like that one question on fetal development that I got wrong isn't going to really affect your score that much. It's more of how well you can read the passages and like pull out information from like the experiments they show you and like the graphs and the data they show you, and like apply it to those questions that they ask you about it. It's less about like what stage does the brain develop, you know. It's like more like, given this table, what can you say about this mutation? Like it's things like that, which is why they're like practice, practice, practice. So I know content review can be very, very overwhelming when you first start, because it's like, oh my god, this is so much information. But like when you actually do the practice, you don't need to know everything.
How many practice exams did you take before you took?
Four, I think. I got 499 on the first one, a 500 on the second one, and that was after like 2 weeks of grinding. I was like, 1 point, really? And then I got a 505, and then I went back down to a 503. Wait, that's way more than 495, 505, 503, 507, 511, and then I got a 510 on the actual. So
Nice.
You know.
Nice.
it is what it is. I wanted higher, but it's okay. I'm not trying to go to
Or
Harvard.
I think yeah, like it puts you in the door. Plus, like I'm sure you have a good GPA and all your experiences are like grade A. And I honestly feel like that should count for more.
Honestly,
because
they're
it's-
starting to look at more, especially after I think with COVID and less people like able to take the MCAT. Like Stanford isn't even requiring the MCAT for this cycle. And like some schools I think are following suit because like a lot of doctors have spoken about it and been like the MCAT does not show how well of a doctor you're going to be. It just shows how much money you have to buy
taking you
resources
have-
Yeah.
to take the
Mhm.
MCAT, because it's like if you don't have any money, you're going to use the free resources, which aren't, as in depth as the ones you pay for, and they're like not cheap. They're like hundreds of thousands of dollars. So it's like it's not how well of a doctor you're going to be. It's how well can you afford to take the MCAT.
Like every school would recognize that because even then it's like you can compare it to the SAT. Personally, I think like the MCAT, as hard as it is, I feel like the content that you've learned and the content that like you gain in college, it's more representative than like an SAT or like a GRE. So when it's funny because people ask me like, wouldn't you rather take a GRE than MCAT? I'm like, in all honesty, I'd rather take an MCAT because like GRE, it's like pointless stuff that's just standardized test knowledge about like, oh, can you remember how to do like trigonometry? Like probably not.
Right.
So it's like
Yeah.
I'd rather take a test that's more representative of like my college experience. Granted, I'm gonna have to grind to like relearn it all because
Yeah.
one it like goes in one ear out the other.
Yeah. The good thing is a lot of doctors are like, and administration is like looking more at holistic applications. Because it used to be for some schools, they had set like a filter for their applicants and they'd be like, if they don't score
higher.
this or above, we don't even look at their application. And that was like, okay, like they could have a 4.0 GPA and like a 500 MCAT score, and then the school just won't let them in. Like I feel like your GPA shows more of your academic work than one singular test. But so hopefully they're starting to change. Hopefully they'll change it more because it's really
Like I book about like grit, and it just like I don't know. I like kind of like those inspirational psych books.
yeah. But I have full faith that you and anyone else watching will do fantastic on the MCAT. All doing great in this class. So
Questions. I know it was like an extra 30 minutes, but
no, it's so fine. I love answering questions about it because
You do today.
I just went through it. It sucked, but Well, if you guys have any other questions, you can always email me too. I don't mind answering. I don't mind sharing my MCAT notes either. That probably is copyright because they're directly from the Princeton Review books, but
We'll keep it between us.
totally gonna email you and hit you up about that
yeah.
because that would be fantastic just to like review.
Well, some of the like diagrams I've shared with the class, like sent in emails, those are from my MCAT notes. I'm just like found them and I'm like, oh, I'll just send these out. So oops, but oh well. But yeah, if you guys have any other questions, please feel free to reach out. I will try to answer. I'm very busy, but I will try.
All right, y'all.
Yeah,
See ya.
Really appreciate the review and like this helpful mini pre-med session.
of
So-
course.
You guys,
Oh yeah, no
Bye.
problem. Yeah, bye.
Well done.
Stop.